Greetings everyone. How can we detect sequencing biases in RNA-Seq data? Are our read counts affected by GC content or sequencing depth? And what is the best way to normalize data before differential expression analysis? Today we will explore EDA-Seq, an R package for exploratory data analysis or EDA, and normalization in RNA-Seq. We will clean and normalize data, not only for DESeq2, but also for edgeR, correcting for GC content and lane effects. Plus, we will use an offset for normalization, a key approach in RNA-Seq analysis. Without further ado, let's get started. Firstly, we upload our libraries. We start by library edseq. This library is specifically designed for RNA-Seq data, pre-processing, normalization, and visualization. And then yeast RNA-Seq. This loads the RNA-Seq datasets, particularly for Saccharomyces. And the third one, BAM views. This provides the example BAM file views from yeast RNA-Seq experiments. And then our data gene level data. This loads a dataset containing gene level RNA-Seq counts. These counts represent the raw data of sequencing reads mapped to specific genes. And then yeast GC. This dataset containing information about the GC content of yeast genes. And finally, data yeast length. This loads a dataset with information on the length of yeast genes. Let's run our code. And now, let's prepare the FASTQ files and their associated metadata for RNA-Seq analysis. Firstly, we write files and we assign it to list.files, file.path, we put system.file, and package equal to yeast RNA-Seq. And then reads, after that pattern, equal to FASTQ, full.names true. Here, list.files, we retrieve the list of FASTQ files from the yeast RNA-Seq package located in the reads directory. And regarding patterns equal FASTQ, here we filter for files with the FASTQ extension. And then file.names equal true to provide the full file path for each FASTQ file. And then names, we put files and we assign it to gsub and we put inside FASTQ base name files. Here we remove the FASTQ extension from the file names using gsub. And regarding the base name files, here we extract just the file names without pass. And then met, and we assign it to data frame conditions equal C rep, and then we put MUT or mutation, and we make it for 2 because we have 2 replicates, and replicate for wild type also 2. Then row.names equal names, and we put files. So we create a metadata data frame to define experimental conditions. For example, conditions replicate mute or mutated 2 and replicate for wild type 2. We specify 2 mutant samples and 2 wild types. And then FASTQ equal to FASTQ file list, and we put files. Then element metadata FASTQ equal to met. We associate the metadata met with the FASTQ object, linking each FASTQ file to its experimental conditions, like mute or wild type. And finally, FASTQ to display the FASTQ file list object. Let's run our code. So as you can see, we have FASTQ file list of length 4 because we have mutant 1F, mutant 2F, wild type 1, wild type 2. Now let's prepare the BAM files and their associated metadata for the downstream analysis. So we write files equal to list.files and we pull the file path, system file. Here we use the package libam views and the files are BAM, pattern BAM, and we put $, and file.names equal true. Then we do the same by names files gsub, and we put inside .bam and base name files. Here we remove the .bam extension from the file names using gsub also. And then gsub to extract the base file name without directories for each BAM file. And then gsub again here to remove everything after the first underscore. As you can see here, we added underscore. So here we remove everything after the first underscore in the file names to capture the general genotype for each sample. And then lane gsub, and we put dot star and geno gsub dot $. Here to remove the last character from the GT variable, leaving the genotype, for example, like mutant or wild type. And then we create the variable called PD. We enter the data frame, geno equal geno that we have created, and lane equal lane, row names equal paste. We put the geno and lane, and for SIP, we enter dot. Here we create a metadata data frame with the genotype geno and the lane for each sample. And for row.names equal paste geno lane SIP, here we set row names by combining genotype and lane. And then BFS equal to BAM file list, and we enter the files, and element metadata BFS equal to PD. And then we display the BFS. Let's run our code. As you can see, we got BAM file list length of 8, and here we have our files. Now let's visualize the BAM file data using a bar plot to examine the distributions or characters of our samples. As one important check for quality control is to look at the total number of reads produced in each lane. The number and the percentage of reads mapped to a reference genome. Low total number of reads might be a symptom of low quality of the input RNA, while a low mapping percentage might indicate poor quality of the reads or low complexity. So, firstly, we create a variable called colors, and we put rep and then RGB, and we put alpha 0.7 and then comma equal 2, and we do it for 4 times, and then we visualize our plot by using bar plot. Then we put the BFS that we have created before here, and the LAS to colors equal colors that we created here. So colors, and then we put RGB, we put the colors. So here is a color, RGB, R equal 1. So here is red, and here is blue, and here is green, and here is between blue and green color. So the bar plot will be around cyan. Let's run our code. As you can see, we got our bar plot. We have 8 bars, 2 per each color. Each bar corresponds to one of the 8 BAM files that we have created in our data. 4 colors representing 4 groups. Each group contains 2 samples. The x-axis here represents the samples, while the y-axis could represent read counts or another measure related to the BAM file data. As you can see, red color for the first 2 samples, ISO WT513E and ISO WT613E. The red bars are among the tallest, reaching about 35,000 on the y-axis, indicating that these samples have one of the highest values among the 8 bars. It indicates that these samples have the highest sequencing depth or expression. But just visualizing BAM files is not enough. As an additional quality check, one can plot the mean per base or per cycle quality of the unmapped or mapped reads in every lane. So let's try to plot our quality scores. We can use plot quality and then we put BFS call equal colors and we make legend to write unique element metadata and we put BFS for 1 and file unique colors. Let's run our code and here we have it. As you can see, we have 4 colors. Red is ISO-WT and we have blue RLP, green SSR, and cyan is XRN. On the x-axis we have the cycle, while the y-axis represents the quality. So as you can see, the red and the blue have the highest quality score, around 70, and then they started to decrease until they get around 45, while the cyan or XRN shows the lowest quality score, which is around 41 or 42. Also, if one is interested in looking more thoroughly at one lane, it is possible to display the per-base distribution of quality scores for each lane and the number of map reads satisfied by chromosome or strand. And in this case, we can also use plot quality BFS and we put here 1 for the first lane and then CEX.axis and we put 0.8. Let's run our code. So as you can see, we have a quality plot for the first BAM file. And that's why here we put 1. On the x-axis, here, we have the sequencing cycles ranging from 0 to 35, while the y-axis, sequence quality, is possibly reflecting the matrices like read quality or base calling accuracy over cycles. We can also create a bar plot for the first BAM file in the BAM file list object. This will allow us to visualize matrices like read counts or coverage across different regions of the genome. So we use bar plot and we put BFS and we put 1. Let's run our code. And here we have it. As you can see, the code generated a bar plot from the first BAM file from the BAM file list object and adjusts the orientation of axis labeled using class argument. The x-axis here contains a feature called SC chromosome 13, which likely refers to a specific region or chromosome, in this case chromosome 13 in the reference genome. While the y-axis here from 0 to 30 or 30,000, it shows that SCR13 or chromosome 13 is about 35,000 here, which corresponds to the number of reads. Now let's read nucleotide distributions. A potential source of bias is related to the sequence composition of the reads. In this case, we can use plot NT frequency and we put BFS 1. NT represents nucleotide frequency distribution. This function generates a plot that shows the nucleotide frequency distribution for all the reads in the specified BAM file. And as we said, NT for nucleotide. Let's run our code. And here we have it. As you can see, x-axis represents the cycles, which are from 0 to 35, while the y-axis for the nucleotide frequency. As you can see, A and T, here we have A, C, G, T, N. A and T, which are the black and the blue here, have the higher frequencies from about 0.25 to 0.3 here. While G and C, which are the green and the red here, have a lower frequency, about 0.2 to 0.25. And for the N, this is for the ambiguous nucleotides, has frequency of 0, meaning no unknown bases were detected. Now we load the gene level dataset, which contains gene expression data from an RNA-Seq experiment. we can use the head function. So first we write the data and then gene level data, and then we can read it by using head. Let's run our code. As you can see, each row here represents a gene identified by its gene ID, while each column represents the expression level or read counts for a specific sample. So mutant 1, mut 2, wild type 1, wild type 2. And as you can see, for YHR here, 055C, it has a count of 0 across all samples, indicating no expression or no reads mapped. Now let's load the yeast length dataset. This contains the length in per base of each gene in the yeast genome. So we do the same, we write data yeast GC and head yeast GC. Let's run our code. As you can see, the gene lengths vary. This contains the GC content, which is the percentage of guanine and cytosine bases for each gene in the yeast genome. We can use also data yeast GC and head yeast GC. For YAL001C, the GC content percentage is about 37%. Here is the same, more or less, and here YAL003W is about 44%, and so on. We can also load the yeast length dataset, which contains the length in base pairs of each gene in the yeast genome. Here. So, as you can see, for example, here, YAL001C, we have 3,483. These are the number of base pairs, while other genes, here for example, contains only 621, and so on. Now we need to filter the non-expressed genes based on their mean. So we can write filter and then apply gene level data. We put 1 function to apply function x, while the mean of x greater than 10. And then table filter and then filter to display the data. So run our code. The genes that have the mean greater than x got true, while the others got false. And now we can also look for the genes that are present in both yeast GC dataset for GC content data and the filtered gene level data dataset, which are the genes with mean expression greater than 10. In this case, common, intersect, names, and we put the data, which are the yeast GC, and for row names, gene level data. And then with length to know how many genes. Let's run our code. As you can see, it's about 4,994. DESeq package provides the Seq ExpressionSet class to store gene counts, lane level information on the sequenced libraries, and gene level feature information. We use the data frame met created in section SecRead for the lane level data. As for the feature data, we use gene length and GC content. So, feature data.frame GC yeast GC, and we put the length yeast length, and then data new Seq ExpressionSet counts equal to as.matrix gene level data, and then we put common feature data equal features common pheno data equal to data.frame conditions. We put the replicates, mutant, and wild type, and row names, column names, and then gene level data. And then we display the data. Let's run our code. So as you can see, the output describes a Seq ExpressionSet here. This containing about 4,994 features or genes for 4 samples, 2 mutant and 2 wild types. Here we have the sample names and labels conditions and so on. The row names of counts and the feature data must match, as well as the row names of the phenodata and the column names of counts, to ensure correct data alignment. The gene expression values can be accessed with counts, sample information with p data, and gene features with f data. Now let's look at our data. As we know, head, and we put counts and data. As you can see, here we have the samples and here we have our genes. We can also look at the sample metadata. So p data, and we put our data. As you can see, it shows the experimental conditions, mutant or wild type, for each sample. So mutant, mutant, wild type, wild type. So instead of mutant 1, mutant 2, mutant, mutant, wild type, wild type. And then we can look at our data. Here we have the GC content, and here we have the length, and here our genes. Now let's use head offset data to view the first few offset values from the Seq ExpressionSet object. Offsets are adjustment factors used in RNA-Seq analysis to account for biases like sequencing depth or gene length. These values help normalize the raw gene expression counts for better comparison across samples. So, head, offset, and then data. So as you can see, the output shows the offset values for each gene across the 4 samples. In this case, the all offset values are zeros. As you can see here, 0, 0, 0, and 0. This suggests that no adjustments or normalization factors have been applied to the data for these genes, or the offsets were not calculated yet. But what about between lane distribution of gene level counts? One of the main considerations when dealing with gene level counts is the difference in count distributions between lanes. Box plot method provides an easy way to produce box plots of the logarithms of the gene counts in each lane. We use box plot function and we put the data, columns, colors from 1 to 4. Let's run our code, and here we have it. As you can see, here is the samples in the x-axis, and the y-axis, it represents the raw gene level counts. And for the larger box plot for wild type 1 and 2, this suggests a wider distribution of counts, higher variability in the number of reads for each gene, and the smaller box plots in the mutant 1 and 2, this indicates less variation in the counts, meaning these samples have more consistent gene level counts across the genes. We can also use mdplot method to produce a mean difference plot of read counts for 2 lanes. So we put mdplot and we put the data C1 and 3. Here we compare the read counts between 2 specific lanes or samples. In this case, lane 1, which is the mutant 1, and lane 3, which is wild type 1. Let's run our code. As you can see, we have on the x-axis the mean, while on the y-axis the difference. This plot is the main difference plot, compares lane 1 mutant type 1 versus lane 3 in wild type 1. X-axis is the average paired values for each pair of mutant 1 and wild type 1, while the y-axis here, for the difference, it represents the difference between the paired value, mutant 1 and wild type 1. But what about the blue shading highlights? The blue shading highlights areas with higher density of points. As you can see, most points are near the dashed line here. These are the points. This is the dashed line. The dashed line represents no difference between 2 measurements. while the black curve here, it indicates that differences slightly increase here as the average values rises, suggesting that higher values may have systematic differences. So in short, mutant 1 and wild type 1 mostly agree, but differences slightly increase with higher measurements. But what about over-dispersion? Although Poisson distribution is a natural and simple way to model count data, it has the limitation of assuming equality of the mean and variance. For this reason, the negative binomial distribution has been proposed as an alternative when the data show over-dispersion. The function mean var plot can be used to check whether the count data are over-dispersed for the Poisson distribution to check whether the count data are over-dispersed. So let's check the first two samples or columns in our data. Let's check. So mean var plot data 1 to 2, same as we said, and log equal true. Let's run our code, and here we have it. So as you can see, this plot compares the mean and variance. Poisson distribution assumes that the mean and variance of the data are equal. But if the variance is much larger than the mean, the data are said to exhibit over-dispersion. And in this case, we should use negative binomial distribution. So as you can see, here's a variance of expression levels for the same genes, while the black line here indicates the expected relationship between mean and variance under Poisson distribution, where variance equal to the mean, while the red line under here represents the observed trend in the data, which accounts for over-dispersion. As we can see, the variance increases with the mean as expected in count-based points deviate from the black line. Here, as you can see, here and here and here, especially at higher mean in this area. And this indicates over-dispersion, and that's why we need to implement negative binomial distribution. Lastly, I would like to mention the blue density areas shows distribution of the points, with darker areas represents regions with higher density of genes, like here, here, and so on. Now let's check all samples in our data. So we can use mean var plot data log equal true and ylm c o and 16. Let's run our code. And here we have it. This reflects that the data set with increased over-dispersion by larger deviation of the red line here from the black line. Note that the mean variance relationship should be examined with replicate lengths only. So for example, conditional on variables expected to contribute to differential expression. For the yeast dataset, it is not surprising to see no evidence of over-dispersion for the two mutant technical replicate lanes. Likewise for the two wild types lanes. However, one expects over-dispersion in the presence of biological variability when considering at once all four mutant and wild type lanes. Now, let's see the dependence of gene level counts on GC content by using bias plot. So, bias plot, data, and GC log equal true. Let's run our code. As you can see, the black line indicates for the mutant type, while the red line for wild type. Here on the x-axis represents the GC content, while on the y-axis represents the gene counts. As you can see, gene counts increase with the GC content up to around 0.40 here. Then plateau or decrease. Mutant samples consistently show higher gene counts compared to wild types. And to show that GC content dependence can bias differential expression analysis, we can produce stratified box plots of the log-fold change of read counts from the two lanes by using the bias box plot method. And again, the same type of the plots can be created for gene length or mapability. So LFC log counts, and we put the data 3.0.1, and we make it minus log counts data, and then bias box plot. Let's run our code. And here we have it. As you can see, here is the bins for the x-axis, which represents the GC content grouped into bins, while the y-axis here, it's for the log-fold change, showing differences in expression between the two lanes. And this is the red line on 0 y-axis because it indicates no bias, so the log-fold change equals 0. As you can see, box plot represents the distribution of LFC or log-fold change values for each GC bin. Most box plots are centered around the red line, suggesting minimal GC-related bias. Now, let's make normalization. In this step, we perform normalization to adjust for various biases that can affect RNA-Seq data, ensuring more accurate comparisons between samples. There are two main types of normalization used, either within lane normalization or between lane normalization. Within lane normalization, it adjusts for biases that occur within a specific lane, such as gene-specific effects, for example, GC content, gene length, and so on. This helps make the counts more comparable for different genes within the same sample. And the second type, or between lane normalization, this adjusts for biases between different lanes, such as differences in sequencing depth or other distributional differences across samples. So, data within or data normalization. Data within lane normalization, we put data and GC, and we put which equal full, and data norm between lane normalization, data within full. Let's run the code. And now let's see how the GC content bias is reduced and the distribution of the counts is the same in each lane. So, bias plot again. Let's run the code. Now, as you can see, it's a big difference. Look here and look here. So as you can see, there is a big difference after normalization. Let's check the box plot of the samples. So box plot, data norm. Let's run our code. As you can see, it's different also. So as you notice, the wild type is very close to the mutant type. I'd like to mention that some authors have argued that it's better to leave the count data unchanged to preserve their sampling properties, and instead use an offset for normalization purposes in the statistical model for read counts. This can be achieved easily using the argument offset in both normalization functions. So here offset true, offset true. One of the main applications of RNA-Seq data is differential expression analysis. The normalized counts, or the original counts and the offset, obtained using the DESeq package, can be supplied to packages such as edgeR or DESeq2 to find differentially expressed genes. So, firstly, edgeR, we can perform differential expression analysis with edgeR based on the original counts by passing an offset to the generalized linear model. So, edgeR, and we write design model.matrix conditions data p-data, and we put data offset, and then DGE list counts equal counts data offset, and y offset offset data offset. Then y equal to estimate disp y and design, and then fit glm fit, we put y and the design, and then glm lrt fit coefficient 2, and finally top tags. I'd like to mention here that even though the data has been normalized using DESeq to correct for biases such as GC content and sequencing depth, we still need edgeR for statistical modeling and inference of differential expression, as edSeq normalization adjusts the data to remove technical biases. But edgeR, or other tools like DESeq2 even, they perform additional steps like modeling the data's dispersion, estimating variance across samples, and testing for statistical significance. Additionally, edgeR accounts for offset like GC content during the statistical modeling process to ensure the results are both biologically meaningful and statistically valid. So, after design, model matrix, and the condition, y DGE list, and then we estimate the dispersion variance or gene expression across samples, which is crucial for detecting the variability in gene expression. Then we fit the GLM or generalized linear model to the data, incorporating the design matrix and estimated dispersion. Then we perform the LRT or likelihood ratio test to compare two conditions and identify significant genes. Now let's run our code. And here we have it. So as you can see, the rows represents to the genes, and here we have the log fold change, log CPM, LR, p-value, FDR, and so on. And also, as we said before, we can apply DESeq2. DDS equal to DESeq dataset from matrix, and we put the count data equal counts data offset, as here we create the DESeq dataset object from the counts data, and then the normalized factors to calculate the normalization factors based on the offset, so offset and data offset. And then normalization factors, DDS equal to norm factors, then DDS, DSeq, and DDS, and then we get the results. Let's run our code. We have the base main, log2 fold change, LFCSE, stat, p-value, and p-adjusted value. DESeq2 automatically performs normalization during its internal workflow. It calculates size factors based on the median ratio method. However, in this case, we are manually adding the normalization factors, as here we put, because we are using the edSeq package for pre-normalization of the data before feeding it into DESeq2. So normalization in edSeq2 perform two types of normalization, within lane normalization and between lane normalization. And after normalization using edSeq2, the resulting counts are adjusted. But we need to ensure that DESeq2 uses the correct normalization factors like offsets so that it accounts for the biases and differences already addressed by DESeq2.
And here we've reached the end of our analysis. Make sure to subscribe to our channel and like the video if you would like to see more content like this. I will leave the links in the description of the video that helped us to do this analysis. If you have any questions or feedback, feel free to drop them in the comments below. Thanks for watching. See you next time.